Start by thanking the organizers for the invitation and especially for the idea of holding this event to celebrate the first decade of these awards, and hopefully it will continue for many more. My work has been concerned with trying to develop genetic, physiological, and pharmacological approaches to studying spinal cord development and disease. And in order to do these kinds of complex experiments, it's best to have a simple model to work with. So the model we work with, we thought ideally should have a spinal cord. Now that's not absolutely required, as we'll hear in the next talk. You don't need to necessarily have a spinal cord to study spinal cord function. But nonetheless, it would be one nice criterion. Another would be able to have easy access to many embryos to be able to do these studies and to have genetic and other tools available. And so the model we've chosen is the zebrafish embryo. And this is a newly fertilized egg. The eggs are about 1 millimeter in diameter, and this is just following the first cell division. So the cell divide every 20 minutes, and I'm going to show you an accelerated view of most of the first day of development. So if I let this movie roll, you'll see division by division. We wind up with a little ball of cells. So this is the blastomere. And in this zone here, at 4.5 hours, there's an inducer. Here it is. It's going to induce the head and tail of the embryo. This is common to all vertebrate embryos. And now you'll see the eyes appear at the top, and you see somatic chunks of muscle along the trunk that develop every 30 minutes. And so at this stage, they start moving for the first time as motor neurons innervate the muscle. So it's a very simple animal. It's transparent, rapid, develops rapidly. You can do an experiment and get the answer the next day, literally. Many technical advantages. Also, the shape of the embryo looks very much like the shape of all other vertebrate embryos, including humans. And not only is there a conservation of gross morphology at these early stages, there's a conservation of the cellular composition and of the genetic mechanisms that generate the cell diversity in the spinal cord. So it's very easy to go from one animal to another and back, as I'll try to show you over the next few minutes. Obviously, later in development, they become highly specialized in their forms. Now, if you look at the zebrafish embryo in more detail, it does have a very simple brain, as does the embryos of most vertebrates at this stage, and the spinal cord lies under these blocks of muscles. Each somite is 50 microns. That's the diameter of a single one of your motor neurons. But this is a functional unit in the fish. They repeat, and each unit contains about a dozen neurons. Now, there are 3 motor neurons that aren't shown here, but as you heard in talks this morning, there's a genetic program that's applicable to fish as well as other vertebrates, where different classes of interneurons, for example, can be identified in the fish, and as in mice, we have transgenic lines that express all of these under different formats. Also, there's 2 types of functional neurons. One is glutamatergic, which is excitatory, and the other is glycinergic. Now, it turns out that glycine is the major inhibitory transmitter of the adult spinal cord, but paradoxically in the embryo it's excitatory, and no one really understands why. And in fact, what we were able to show is that if you block glycinergic activity in the embryo, you lose half of the interneurons, but not the other cell types. And so that was the work that was recognized with the Turnbull Award. And what we've done in more recent years is look at this in more detail. I'm going to summarize quickly that there's a proliferation and migration of progenitors, but it's their differentiation into neurons that seems to be regulated in part by the actions of transmitters that will be excitatory in the embryo and stop being so later in the adult. The work we've done over the last few years is summarized in this slide, where we think that glycine acts in a paracrine manner as a circulating agent that activates channels that allow chloride ions to leave the cell. These negatively charged ions leave the cell and depolarize it, and as a consequence, calcium channels open, and these activate a signaling pathway that's involved in progenitor differentiation. So I'm not going to go into this in detail. I thought I'd also try to present to you some work that's more related to your interests, if not in spinal cord diseases, at least in spinal cord function and dysfunction. And so for this work, we take a following simple approach. We can inject a construct that's called a morpholino. It mimics an oligonucleotide, so we can target a gene with this. We inject these by hand into the eggs, they diffuse into the cells, and they'll inhibit the zebrafish gene. Now, if we find a gene that bears a mutation in humans, if we find its homolog in the fish, if we knock down, as the terminology goes, the fish gene and get a phenotype, an abnormality in its development, then we can co-inject the human RNA to see whether the human gene can compensate for the fish gene's function. And that happens in over half of the human genes that we've tested. Now the interesting part is, if the human gene works, we can compare the normal gene with variants that are found by genomic studies. And so the point is, are these variants that are benign, or are they actually pathogenic mutations that cause a disease? And you can't guess that necessarily from the biochemistry. You have to have some kind of in vivo readout to get this, and that's the simple goal we have with the zebrafish. So here's one example. This is a gene that was recently discovered. It's an RNA binding gene implicated in amyotrophic lateral sclerosis, or ALS, or as it's known in the United States, Lou Gehrig's disease, for the baseball player who thought to die of this disease. The disease is untreatable, and the mutations in these genes don't alter any of the known biochemical functions. However, as I'll show you, it does have an in vivo effect. So here is a non-transgenic sibling of one of these transgenic fish, and when we touch it, it swims away quite briskly. If we express the human gene bearing a mutation that causes ALS under a particular genetic construct, what we can see is when we touch the fish, it responds, but it doesn't swim very well. Now, I'm not saying this is ALS in a zebrafish, but it is an ALS-related mutation that has a phenotype that shows a motor deficit, and there are motor neuron deficits as well, but I'm not going to go into detail. So this is the basic assay that one can develop. Now, what we can do in addition is we can look at several genes at the same time. You may have heard that over the last 3 years there's been an explosion in the genetics of ALS. We've gone from one gene, SOD1, to 18 genes now implicated in ALS. How do they interact, and how do you study that interaction? I'm only going to show you one small example. This is the knockdown or disactivation of the zebrafish gene that we rescue with the human gene. And here's a second RNA binding gene called FUS, which can also rescue it. So both these genes act on restoring this activity. On the other hand, if we knock down the FUS gene, only its human homologue is capable of restoring the activity, and not this other TRDBP gene. One conclusion is that FUS lies downstream genetically from TDP. So there's a beginning of a network, which we think of as in terms of this ALS matrix, where FUS can rescue anything that's upstream of it, and TDP can only rescue itself. We've now extended this to several other genes, and I'm only going to show you in a cartoon summary the thought that the mutations of TDP cause a loss of their function as the proteins leave the nucleus, and they become toxic in the cytoplasm, just as SOD1 becomes toxic. We're acting through different pathways, suggesting that there may be different etiologies to these diseases, and this fits in well with recent biochemical as well as clinical data that there may be multiple causes now of ALS. Now, we also thought of using this as a developmental model in the context of developmental disorders. So this is a project that we call Synapse to Disease, and the hypothesis is an old one, that synaptic genes may be important in disorders like autism and schizophrenia, where it's the function rather than the morphology that seems to be aberrant in the patients. And the idea that we came in that was novel is that perhaps the mutations may be de novo. So the point is that it's been very difficult to find genes that are causative of autism and schizophrenia from classical population genetic studies. Could it be that, like in cancer, many of the patients have mutations that they do not inherit from their parents, but that occurred during very early development? That's 90% of the causes of cancer, but they haven't been demonstrated in brain diseases. And the only way to do this is to go in and sequence these genes on a very large scale. And so this is work that we called Neuromics. It was an email title that I sent to my collaborators many years ago proposing this idea. But it was largely performed by Guy Rouleau and Jacques Michaud, who were neurologists and geneticists at the Saint-Justin Hospital in Montreal. So basically we've sequenced several hundred, close to 1,000 synaptic genes in several hundred patients affected with schizophrenia or autism. So this is by sequencing the genes, looking for the variants, and now the key question is, are these variants pathogenic or not? So we test them in my lab in zebrafish, other labs use them in Drosophila or in C. elegans, as well as in cultured neurons. And the conclusion from our analysis of the first few hundred genes, so we looked at 406 synaptic genes and found 21 de novo mutations. So this means that in 4% of the patient cohort, we found de novo mutations which we could show were pathogenic in various models. But 400 synaptic genes is probably only a tenth of all the genes required to make a synapse, which by extrapolations means that it may be that as many as half the cases of autism and schizophrenia may be due to de novo mutations that are not inherited, and that's why they're very difficult to detect by other approaches. Now, this is a list of diseases we've looked at. I'm not going to go through them all. These are genes implicated in autism and schizophrenia. These are genes that I mentioned in the context of ALS. We also collaborated with Patrick Cossette at the University of Montreal. He and his clinical colleagues have discovered a new disorder called MEDNIC. It touches a whole variety of organs, from skin to the brain. It's a clathrin adapter protein that's expressed in every cell, so it's a widespread mechanism. And we found the mutation, we validated it in the zebrafish, and we finally got this paper published. And so I told my wife, we've discovered this new disease. And she says to me, what are you doing discovering new diseases? I mean, there's enough diseases out there already. Why don't you do something about trying to find a cure for something instead? So I listened to my wife's advice a lot. After 30 years, she's been more often right than wrong, certainly more so than me. And so we started thinking, how can we use our model to go from the discovery of causes of diseases to trying to find cures? I'm going to show you one example. So the idea is that we can take embryos from our transgenic fish and plate them into these little tiny wells. There's 96 wells on one of these plates, and the plates are pre-filled by a robot with one of many thousand different compounds. Now, we've done this in zebrafish, and we've done it in parallel with Alex Parker in Montreal using a C. elegans model that has a very similar motor deficit. And we've gone through over 3,000 molecules over the last summer, and we found 15 that seem to improve our phenotype. So we're not looking for compounds that are going to make things worse. We're looking for the hard part, compounds that will make these behaviors better. And I'm going to show you one example here. So this is a normal zebrafish embryo that's just hatched, and when we touch it, as you expect, it swims away and is being tracked by a computer. So the line in blue allows us to reanalyze the data more quantitatively. Now we take the fish that's expressing the TRDP mutation that's related to ALS. When we touch it, it responds, but it barely moves. And now